Now that we have gone over the whole pipeline in the first level of detail, let's dig a little deeper. We will begin with the FASTQ file format. As previously stated, FASTQ files store biological sequence data, with each entry consisting of exactly 4 lines: the read ID, the nucleotide sequence, an irrelevant line, and the quality scores of the nucleotide sequence. Let's talk about the read ID. Each read ID is, in theory, unique. No two reads in existence should have the same ID, since read IDs are normally generated by a combination of factors, such as the ID of the sequencing machine, the time and date of the sequencing run, flow cell coordinates, and so on. Read IDs are usually preceded by an @ character. The nucleotide sequence contained in the second line is fairly straightforward. It is a sequence of letters, with each letter corresponding to a nucleotide. A for adenosine, C for cytosine, G for guanine, and T for thymine. If the nucleotides at a given position is indeterminate, a placeholder will be used. For DNA sequences, this is typically an N. FASTQ files can also store amino acid sequences. I won't go over the one-letter codes for all 20 amino acid residues, but the placeholder letter for those sequences is typically an X. The third line is a bit of an oddity, in that, for the most part, it doesn't seem to have any uses. It usually begins with a plus character and can otherwise be empty. Sometimes the third line will begin with a plus character and then contain the same information as the first line of the entry, minus the @ symbol. Not much to see here. The fourth line is where it gets interesting. For quality scores, FASTQ files use a particular system. Instead of recording a number for each position in a human-readable format, FASTQ files use one character per position. Each character represents a different number, and those numbers range from 0 to over 40. It is done this way in order to reduce the file size and to make the file easier to compress. Because such files are mainly processed by computers, making the raw data easy for humans to read was a low priority when the FASTQ file format was being designed. So each character represents a number, but what do those numbers mean? The quality score numbers are known as Phred Scores, and they indicate how likely it is that a given base call is incorrect. In most cases, Phred Scores are calculated using the formula Q equals -10 times log P, where Q is the Phred score, P is the probability of the call being incorrect, and log is in base 10. In specific cases, such as the Solexa pipeline prior to version 1.3, the formula is Q equals -10 times log, open brackets, P over 1 minus P, close brackets, where Q is the Phred score, P is the probability of the call being incorrect, and log is in base 10. The key take-home in regards to quality scores is the higher, the better. A Phred score of 10 indicates a 1 in 10 probability of an incorrect base call, or an accuracy of 90%. A Phred score of 20 indicates a 1 in 100 probability of an incorrect base call, or an accuracy of 99%. A Phred score of 30 indicates an accuracy of 99.9%, and so on. If you are really interested, there is a Phred score conversion table in the appendices video. Phred score conversion tables are also very easy to Google. It is important to note that there are at least 2 Phred systems used in FASTQ files. Phred 33 begins at ASCII character 33, with the exclamation mark symbol corresponding to a score of 0, while Phred 64 begins at ASCII base 64, with the @ symbol corresponding to a score of 0. Therefore, it is important to first ascertain the Phred system being used before commencing analysis. The presence of certain characters would indicate that a file cannot possibly be using a Phred 64 system. They are shown on screen, but I won't read them all out loud. If none of these characters are present in the quality scores of a FASTQ file, then that would be a strong indicator that the file uses Phred 64. If a FASTQ file uses Phred 33 but doesn't contain any of these characters, it would mean that every single base call, without fail, had an accuracy of 99.37% or higher, which is unlikely.